Good morning. Welcome to the 5th International Conference on Applied Zoology 2022. I am Muhammad Kabir, MPhil Scholar from Government College University, Faisalabad. I am going to moderate this session on the behalf of AZSP, the Applied Zoological Society of Pakistan, including talks from various fields of sciences such as biodiversity, ecology, biotechnology, and molecular biology. Let's make a start of today's session. This session will include 8 abstracts in total. Our first presenter is Dr. Malik Sadullah from Government College University, Faisalabad. He will present his work on a novel distachionate from Brinia distachia treats inflammations by modulating COX-2 and inflammatory cytokines in rat liver tissues. Please, Dr. Malik Sadullah.
Bismillahirrahmanirrahim. A novel distachionate treats inflammations by modulation COX-2 and inflammatory cytokinase in rat liver tissue from Brinia distachia. First of all, introduction. Importance of plants in Islam. In Holy Quran, it is said, "Your God is that who has made different kinds of orchards and gardens for you. Those have colorful crops of Phoenix dactylifera, Olea europaea, and Ponica granatum." Olea europaea. Olea europaea, that is olive, has found many references in Holy Quran and Hadith. In Hadith, it is said, "Eat the olive oil and apply it on the body. It cured 70 diseases." Kalwanji. According to Holy Prophet, peace be upon him, a regular use of kalwanji can cure every disease except death. Here are some important drugs of plant origin. First of all, aspirin. Chemically, it is salicylate. It has analgesic, anti-inflammatory, and antipyretic activity and obtained from Salix alba. The second one is atropine. Chemically, it is alkaloid. It has pupil dilator activity and obtained from Atropa belladonna. The third one is cocaine. Chemically, it is terpene alkaloid. Medicinal property, it has local anesthetic activity and obtained from Erythroxylum coca. Importance of natural products in modern medicine. Compounds from natural sources such as plants and animals play a significant role in modern medicine. They provide a number of extremely useful drugs that are difficult, if not possible, to produce commercially by synthetic means. These include such diverse groups of compounds such as alkaloid, morphine, codeine, vincristine, vinblastine, taxol, atropine, and hyoscine. Plant collection, identification, and extraction. Brinia distachia was collected from peripheries of Lahore, Pakistan in August 2019. The plant was authenticated as Brinia distachia by Professor Dr. Altaf Ahmed Dasti, taxonomist, Institute of Pure and Applied Biology, Bahauddin Zakariya, University, Multan, Pakistan. The voucher specimen has been deposited in herbarium. Dried and grinded plant material of Brinia distachia was extracted by using different organic solvents such as dichloromethane and methanol. Through a successive maceration and filtration, the filtrates were concentrated by using the rotary evaporator. Result of extraction. The part used was aerial part. 900 grams of product plant were used. Organic solvent used was dichloromethane and methanol. 12 grams extract obtained from dichloromethane and 19 grams of extract obtained from methanol. Different phytochemical test was also performed. The result of phytochemical screening shows that saponin, steroids, and triterpenoids, flavonoids, cardiac glycoside, and tannins were present, while alkaloids and anthraquinones were absent. Here is the Hplc spectrum. The Hplc spectrum shows that quercetin, gallic acid, paracomeric acid, and synapic acid are present in Brinia distachia. Metal analysis of Brinia distachia was also performed. It shows that many metals such as iron, manganese, potassium, zinc, sodium, and magnesium are also present in the extract of Brinia distachia. Here is anti-inflammatory assay guided isolation. By using open column chromatography, we purify the compound by repeated column using different mobile phases. The schematic diagram of purification is shown here. We elucidate the structure of pure compound by using different spectroscopic methods. These are the spectra of our pure compound. Physical parameters. It was a yellowish powder. Quantity obtained was 9-10 mg. The melting point was 175 to 185 degrees Celsius. UV lambda max was 230-360 nanometer. IR was conducted by KBR method. Proton NMR by using 600 megahertz. C13 NMR, EIMS, and molecular weight and molecular formula was obtained by HREIMS. Here is the chemical structure of distactionate. Biological activities. Different biological activities were also performed. First of all, wound healing potential of Brenia distachia methanol extract by Axion wound healing model. 10% cream of Brenia distachia methanol extract, 20% cream of Brenia distachia methanol extract, and novel distachionate was used against the standard contractobex. The novel distachionate shows a significant result against the standard contractobex. The second one was burn wound healing model. Here is the same 10% cream, 20% cream, novel distachionate, and the standard contractobex was also used. The novel distachionate shows a significant result against the standard contractobex. Carrageen Pa edema model. In this model, saline Brenia distachia methanol extract distachionate was used against the standard indomethacin. The novel distachionate shows a significant result against the standard indomethacin. Cotton pellet edema model. Same as the saline Brenia distachia methanol extract distachionate and the standard indomethacin was used, while the distachionate shows a significant result against the standard indomethacin. Histopathological result. Histopathological analysis of liver, heart, lung, kidney, and spleen is shown here. Liver shows a mild inflammation which was significantly reduced after treatment with Brenia distachia methanol extract. No renal and cardiotoxic activity was observed. Here is a molecular docking. Molecular docking was performed for confirmation of target size. It shows confirmed that novel distachionate treats inflammation by modulation COX-2 and inhibited cytokinase in rat liver tissue from Brenia distachia. Here are the results obtained from docking. DPPH radical scavenging and acetylcholine esterase inhibition activities was also performed. Brenia distachia methanol extract and novel distachionate was taken against the standard. In DPPH activity, quercetin was used as standard and distachionate shows a significant result against the standard. In acetylcholine esterase inhibition activity, Isolene was used as a standard and distachionate also shows a significant activity against the standard. Conclusion. In conclusion, current study isolation of new compound as 2-amino-678 trimethoxy 4A 56789 hexahydro 2-hydroxybenzo aniline 3-ol 3,4,5 trimethoxybenzoate from methanol extract of Brenia distachia isolated compound showed a prominent anti-inflammatory and antioxidant activities which gives a clue about its immunomodulatory attributes. Molecular docking studies show that this distachionate X inflammation by modulation COX-2 and inflammatory cytokinase in rat liver tissue. Further studies to standardize the methanolic extract and long-term toxicological studies are warranted to establish the therapeutic potential of this novel compound. Here is the schematic diagram of the activity of distachionate. Distachionate cure the acute inflammation and chronic inflammation. It does not affect the normal cells. Thank you for your attention.
Thank you, Dr. Malik Sadullah from Government College University, Faisalabad, for such a ingratiating talk. Our next presenter is Dr. Muhammad Kabir from University of Sargodha. He will share his research work on impacts of anthropogenic activities on environment, a critical review. Please, Dr. Muhammad Kabir.
Bismillahirrahmanirrahim. Respected Chair, Co-Chair and audience, Assalamu alaikum. Good morning, afternoon and evening according to different regions of the world. I am Dr. Muhammad Kabir, serving as an Assistant Professor and in charge at Department of Biological Sciences, Tharj University, Bakhar, Punjab, Pakistan. First of all, I pay my thanks to the organizing committee of 5th International Conference on Applied Zoology 2022 who gave me a chance of presentation in this international conference. Audience, today the topic of my presentation is Impacts of Anthropogenic Activity on Environment, a critical review. As we know that environment is consist on three major components, that is air, water and soil, and all these components of environments are degrading due to anthropogenic activity that includes the urbanization, industrialization, transportation and deforestation. So the presentation layout includes the problem, that is anthropogenic activities causing the environmental pollution, and its solution is the plantation, importance of trees economically, pollution, pollutant types and issue, plantation activities, solution for pollution, environmental calendar, conclusion and recommendation, and at the end I will show you some certificates and publication of 2022. Muscosol says a tree that lives for 50 years generates a $$ worth of oxygen, recycles huge quantity of $$ worth of soil fertility, facilitates $$ worth of soil erosion control, creates a huge quantity of $$ worth of the air pollution control, and provides $$ worth of shelter for insect, birds and animals. Besides, it provides flowers, fruit and seed. Our net loss is worth more than 57,381 when one tree falls are affected by environmental pollution. Therefore, a humble request to all humanity on the surface of earth is that think before you cut a tree. What is the pollution, pollutant types and issues? An undesirable change in physical, chemical and biological current harmfully effects to the living organism is known as the pollution. The substances which cause the pollution are known as the pollutant, or simply we can say the residue of things we make, use and throw away are known as the pollutant. What is the issue? We use the resources extensively and then are not responsible for the consequences. In the picture you can see that it is our common practice that most of the industrial waste are dumped outside the industrial wall, and then by their burning different types of pollutants are being discharged into all components of the environment including air, water and soil. When we dump the industrial waste outside the industrial wall, they can cause the pollution into the air, into the water and into the soil. And as we know that soil is the basic medium for the plant growth, and if the soil is polluted, it can affect the growth, development of the plants. And plants are the producer on the surface of earth. When plants will take the pollutant from the polluted soil, then this pollutant will pass into the whole food chain including the human body. Another anthropogenic activity is the cutting of tree, deforestation. We cut the tree and then by their burning different pollutants are being discharged into the environment. Industrial activity are also on the large scale to support the luxury life. So there is a need to think the clean technology, the revolutionary technology, so that we can keep our environment clean and green. So the solution for pollution is plants act as a natural lungs. As we know that plants are the producer on the surface of earth. They carry out the process of photosynthesis. As a result of photosynthesis, they take up the carbon dioxide from the environment and release oxygen. This oxygen is necessary for the respiratory activity of all the living organisms. And carbon dioxide concentration is increasing day by day due to the anthropogenic activity, which is resulting into different phenomena like global warming, climate change, melting of glacier, floods and other such activity which are taking place on the surface of earth. Plant a tree and get oxygen for free. Plants are natural purifiers of the environment. It is our duty to save environment's beauty. Let us go green to get our globe free. So green revolution is the best solution to arrest the environmental pollution. In the next slide, you can see that in our university campus, entrance gates are also made by different plant species. We have planted a large number of trees in earthen pots in front of our academic building. Our staff, students, and faculty members arrange different walks and talks regarding to the plantation activity in collaborations with district administration, divisional forest officer, and worthy deputy commissioner. And in the last portion of the slide, you can see that our worthy director is planting a tree in front of the academic building. So there is a need to think about the environmental calendar or the clean and green environment. As we know that World Environment Day falls on the 5th of the June, World Population Day on the 11th of July, International Plantation Day on 18th of August, Ozone Week on the 16th to 23rd of the September, and similarly Wildlife Week on the 1st to 7th of October, and all the other important events of the environmental calendar are mentioned here in this slide. And most of us are not the eat today, and we are not sincere regarding to the environment. So conclusion, if habitat population growth and construction of new industrial structures, transportation and urbanization activities go on without realizing the importance of trees, then probably there won't be more destructive changes in all components of environment in near future. These environmental changes are harmfully affecting the habitat of living organisms on the surface of Earth. So be a part of the solution, not environmental pollution. The home message for all the participants, which is my request, that each one must at least plant one. In the next slide, you can see that industrial activities are being establishing on the large scale in order to fulfill the luxury human life. And these industrial activities are polluting to the air, to the soil, and their pollutants after passing from the plant body are entering into the human. Next portion of the slide, you can see that without the tree, the temperature on the surface of Earth is increasing, and with the tree, this temperature is under the control up to some extent. If tree gave off the Wi-Fi signal, we would be planting so many trees, we would probably save the planet. Too bad, they only produce the oxygen which we breathe and food which we eat. Join the green revolution and stop the environmental pollution. Here you can see the different certificates which our university has obtained from the UI Green Matrix, and in the second portion, the certificates are regarding to the Research Productivity Award, which I have obtained from Pakistan Council of Science and Technology during 2012 and 11. These are the publication of 2022 in different international peer-reviewed journal. It means that I have playing my role regarding to the environment. So save the environment to save yourself. Thank you for your attention.
Thank you, Dr. Muhammad Kabir, for such a productive presentation. Now it's time for our third presenter of this session, that is Dr. Muhammad Tayyab from University of Veterinary and Animal Sciences, Lahore. His title of presentation is Recombinant DNA Technology for the Production of Industrially Important Recombinant Enzymes. Please, Dr. Muhammad Tayyab from University of Veterinary and Animal Sciences, Lahore.
Bismillahirrahmanirrahim. So first of all, I am thankful to the organizers for providing me the opportunity to present and to contribute in this conference. So I am Dr. Muhammad Tayyab, serving as Associate Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. So the topic of today's talk is recombinant DNA technology for the production of industrially important recombinant enzymes. So I want to divide this topic into two parts. Part one, that is actually the enzymes, which are important for industrial point of view. And number two, the second part is recombinant DNA technology. So let's talk about these two portions one by one. So if we talk about enzymes, so majority of enzymes are actually protein in nature. And we know actually proteins are very important biomolecules. So particularly when you are talking about enzymes, so enzymes are very important. We can see here a variety of applications. These are actually a few of examples, like these can be utilized as biocatalyst in research, in diagnostics, in food and beverage industry, in animal feed industry, pharmaceutical industry, biofuel, and detergent industry. We can have many more applications of enzymes. So here we have some of the examples of enzymes, like for example, we can see here in front of us like glucomylase, glucose oxidase, pectinase, recombinase, endonuclease, polymerase, protease, phytase, lipase, and many more. These are few of the examples of enzymes which are important for the industry. And if we talk about the production of enzymes, basically, commonly there are two strategies which are being followed for the production of enzymes. Number one, that is called fermentation technology, and number two, that is called recombinant DNA technology. In case of fermentation technology, we are going to utilize the wild type strain for the production of some special products, some enzymes. Like for example, here we can see, so we must have actually the inoculum of the wild type microbe, and we are going to provide it to the fermentation medium, and this medium is actually having various nutrients which are being utilized by the microbe for the production of intracellular or extracellular enzymes. It depends on the microbe and its ability to produce various variety of enzymes. So the enzymes which are being produced as a result of this fermentation process, that can be purified and characterized later on so that you can have the final product. Whereas in case of recombinant DNA technology, that is the advanced form of technology which is being utilized nowadays for the production of recombinant enzymes. Before going towards the recombinant DNA technology, let's have a little description about a comparative analysis of fermentation technology versus recombinant DNA technology. So in case of fermentation technology, basically we are going to utilize the wild type cells for the production of enzymes. But in case of fermentation technology, if we are interested in the production of thermostable enzymes, definitely its source will be hyperthermophilic microbes. And we know that hyperthermophilic microbes, these can be grown beyond 90 degrees Celsius. And in case of fermentation technology, it requires the maintenance of 90 degrees Celsius for each batch of microbe so that it can grow and can produce desired amount of enzymes. But the thing is that Pakistan is deficient actually in fuels, in fossil fuels. So this is not a good idea to utilize fermentation technology for the production of thermostable enzymes because it will increase the production cost of the enzyme. And secondly, the yield is low. So overall, we can say that industry, as you know, that industry requires enzyme production in tons and tons, means a large quantity of enzyme is required when you're talking about the application of enzymes in industry. So it is difficult to produce these higher level of enzymes by fermentation technology at economic cost. So when you are going to produce the same enzyme by recombinant DNA technology, so you can say that we just need to grow the microbe once at its desired temperature. Once you have the microbial growth, and might be 100 ml of this microbial growth will be sufficient. So we have to utilize that microbe for the isolation of genomic DNA. Once we have the genomic DNA, we can utilize it for cloning of your desired gene of interest in specific vectors, so that you can have the production of enzyme. And the important thing is that if you are going to utilize some mesophilic microbial strain for the production of as the expression host, so that can grow simply at 37 degrees Celsius. And we know in Pakistan, most of the year we are having this temperature, around this temperature. So enzyme production that is going on at 37 degrees Celsius in case of recombinant DNA technology. But the produced enzyme will have the same characteristics as was produced by the wild type strain. So this means recombinant DNA technology that is most appropriate and suitable for the production of larger quantities of enzymes. So here we have actually a brief description about the recombinant DNA technology. So I'm going to share actually the recombinant DNA technology for the production of enzymes which are required in the poultry feed industry. So there are some of the steps which are important regarding the production of recombinant enzymes. Like, for example, so first of all, we must have actually the hyperthermophile. So the hyperthermophilic strain that will be actually required for the isolation of genomic DNA. Once we have the genomic DNA, we can utilize that genomic DNA for the amplification of the gene of interest by the polymerase chain reaction. Then once we have actually the amplified PCR product, we can utilize these products. Like, for example, we need actually phytase, cellulase, xylanase, and protease. These are a few of enzymes which are important for the supplementation of poultry feed. So these enzymes, once the genes has been amplified by PCR, then these can be cloned separately in the cloning and expression vectors, and the presence of gene in the vector can be confirmed by plasmid isolation and restriction analysis. So once we have confirmed that our gene of interest has been ligated in the vector, so then this recombinant vector can be utilized for the transformation of E. coli BL21 codon plus cells, for example, that is actually can serve as expression host. Then once we have the cells which are having the recombinant vector, so these cells can be utilized for the production of recombinant enzymes. And these recombinant enzymes, after the production, we can optimize actually the conditions for the production of these enzymes. And once we have the characterized enzymes, we can actually utilize these enzymes for the supplementation of poultry feed. And then ultimately by poultry trials, we can get information about the impact of these enzymes on the growth of poultry. So this was a very simple discussion about various steps important in the recombinant DNA technology for the production of recombinant enzymes. So as a conclusion, we can say that recombinant DNA technology that is actually a latest and important technology for the production, for the bulk production of your desired enzymes at economic level. So thank you. If you have some question, you are welcome. Thank you.
Thank you, Dr. Muhammad Tayyab, for such an astounding presentation. Our fourth presenter of this session is Dr. Shabnur Iqbal from Government College University, Faisalabad. He will share his research views on virtual screening of some selected compounds of Artemisia absinthium to assess anti-inflammatory effect against tumor necrosis factor, cyclooxygenase 2 enzyme, and nuclear factor kappa B. Please, Dr. Shabnur Iqbal from Government College University, Faisalabad.
Presenting for the 5th International Conference on Applied Zoology. And my name is Dr. Shahzad Irfan. I'm teaching assistant in Department of Zoology, Government College University, Islamabad. My study entitled Virtual Screening of some selected compounds of Artemisia absinthium to assess anti-inflammatory effect against human necrosis factor, cyclooxygenase enzyme, and nuclear factor kappa B. Artemisia absinthium is very important medicinal species and formally described in medieval Europe as most important master against all devastation. And it is also very important traditional medicinal plant in Asia and used against very different ailments like gastrointestinal ailment, to treat the helminthiasis, anemia, insomnia, bladder diseases, and to reduce the wound inflammation and healing. It is important to discuss the molecular docking as virtual screening is based on the one of the technique of the molecular docking. Docking studies are actually computational techniques for the exploration of the possible binding modes of substrate to a given receptor or enzyme. In molecular docking, there are two types of interaction were studied: protein ligand interaction and protein-protein interaction. In our study, we use the protein ligand interaction, and ligands are structure that binds to binding site of the receptor, while the receptor contains the active site, uh, and the binding site of the set of amino acids that physically interact with the ligands. The ligand and protein interaction based on the two theories that are the lock-and key theory and loose fit theory. Interlocking key theory was proposed by the investigator. According to him, the lock is the enzyme and the key is the substrate. And just like that, only the correctly sized key fits into the hole of the keyhole of the lock. Just like that, the substrate of particular size attached to enzyme with same size and structure. In this diagram, you can see that the substrate structure is different, so it cannot bind with the binding site of the enzyme with different structure and shape. So the next, you can see that the binding site of the enzyme and substrate have the same shape and structure, so they can attach and make the product. Loose-fit theory explains certain molecules bind with the enzyme but do not react because enzyme sometimes distorted due to molecules or other some molecules are too small to produce the proper alignment, and only proper substrate is capable of producing proper alignment with the active side of the enzyme. These are some software of molecular docking that are Sanjeevani, Gold, Autodock, ChemDock, Hex protein docking, Gram protein docking. In our study, we use the Autodock methodology. In the best study, we choose 14 compound of the Artemisia absinthium, and the compound was selected based on the previous reported study. This is a schematic view of our study, and that the first step we select the ligands and receptor, and then we prepared in the software of the discovery visualizer, and in the Pyrex software we process the receptor ligand docking, and at the last we studied the binding interaction of the receptor and ligand. For the preparation of the receptor, we use the BioVia Discovery Studio, and for this we remove the in this software we remove the heteroatoms, bonded ligands, water molecules from the protein chains. Only protein with the particular chain and active site were saved in the form of PDB file, and this is the software of Discovery Studio Visualizer. In the Pyrex software, we upload our desired targeted receptor and ligands, and you can see the software here. And the molecule side, we uploaded the ligands and receptor, and then we start the Vina wizard. And in the gray area, we get our 3D structure of the ligands and receptor, and then we run the Autodock Vina and get our result of docking score. This is the list of Artemisia absinthium compound, and they are actually downloaded from the PubChem, and in the form of STF files, and these are the listed their compound IDs. These are the 3D structure of the our targeted receptors, and they were downloaded from the protein data bank, and by using their PDB IDs results. In first result, you can see that the receptor ligand interaction of NF-kB with our targeted ligands or compound of the Artemisia xanthium, and you can see that the best result were obtained between the NF-kB and secretiole, and that give the best binding energy, or you can say binding affinity. And in the next, these are the binding traction of TNF alpha with our target ligands, and you can see that the secretiole give best docking score with TNF alpha, and out of the 14, almost all compound give the binding traction with TNF alpha. In the COX-2 and ligand interaction, we can see that the best result same given by the secretiole, and out of the 14 compounds, 13 give the binding affinities. Best result you can see that given by the secretiole. These are the 3D structure of the ligand binding interaction, and we use the uh, discovery studio visualizer to make these interaction, and we only visualize those structure that give the best or better binding affinities with COX-2, and these are the interacting residues of the COX-2 and the compounds, and residues are actually amino acid of at the active site, and where they from which they interact with the ligands. These are the 3D structure of 3D structure of the NFKB and the compounds. Next, you can see the interacting residues, and these are the interacting structure of the NFKB with the molecule compounds, and that give the best binding affinities. And these are their interacting residues that are the amino acid you already discussed. And these structure are basically we processed in the discovery studio visualizer, and in first step we upload our receptors, and then we define our ligand, and after uploading the ligand and after the defining, we get our interaction structure, interacting structure that you can see, and then at the last we get labeled this interacting structure and results. Out of the 14 compound of the Artemisia xanthium, TNF exhibited binding affinities with all compounds, and best score were given by secretiole, and its docking score was minus 0.7 kilocalories per mole. And out of the 14 compounds, COX-2 was expressed good binding affinities with 13, and except mycin, and my minimum binding energy was given by the secretiole, and that was a good binding affinity and docking score. And NFKB also give good binding affinities with the secretiole, and at the last conclusion, the virtual molecule screening was helped to reveal absentia. Artemisia xanthium has binding affinities, has anti-inflammatory effect into phytoconstituents, and among 14 compounds, secretiole was found most effective anti-inflammatory agent owing to good binding affinities with TNF alpha, COX-2, and NFKB, and that might use in future study to elaborate their possible anti-inflammatory pathway. Study also concluded that the COX-2 and TNF alpha are important receptors that are attributed to inflammation and might be down-regulated by secretiole of that was the compound of the Artemisia xanthium. Thank you very much for mention.
Thank you, Dr. Shabnur Iqbal, for delivering such a delightful presentation. Our fifth presenter of this session is Dr. Shahzad Irfan from Government College University, Faisalabad. His title of presentation is Temporal Assessment of Hepatic Expression CREP Carbohydrate Response Elemental Binding Protein in alloxan-induced hyperglycemic adult rats. Please, Dr. Shahzad Irfan from Government College University, Faisalabad.
Bismillahirrahmanirrahim. My name is Dr. Shahzad Irfan. I'm presenting my abstract, the 5th International Conference on Applied Zoology 2022, organized by the Applied Zoological Society of Pakistan in collaboration with Khwaja Farid University of Engineering and Information Technology, Rahim Khan, Pakistan. The title of my presentation is the Temporal Assessment of Hepatic Expression of Carbohydrate Response Element Binding Protein in alloxan-induced hyperglycemic adult rats. So the first section is the introduction. Carbohydrate response element binding protein, or CREP, is a transcription factor and is activated by the carbohydrate metabolites. And after its activation, it results in the upregulation of different glycolytic and lipogenic genes in the liver. CREB is expressed at high levels in the hepatocytes, in the beta cells, adipose, white and brown adipose tissue, and the muscles. And lower expression levels have been observed in the physiologically high glucose levels induce CREB acetylation. This acetylation leads to an increase in the binding affinity, chemical stability, and transcriptional activity of the CREB. Glucose 6-phosphate, or G6P, is the primary carbohydrate metabolite, which directly binds to different conserved sites on the CREB. And these conserved sites are comprised of low glucose inhibitory domain, or the LID, and glucose response activation conserved element, or the GRACE. And both of these domains are collectively known as the glucose sensing module of the CREB, which implies that the glucose as an important regulator of the CREB. Now, if we look at the isoforms, the CREB has two different isoforms, alpha and beta. The alpha is the classical or the canonical isoform, whereas the CREB beta is expressed by an alternative promoter. The main difference between the alpha and beta domains is that the beta domain lacks the low inhibitory glucose domain, which means that at the low glucose level, the transcriptional activity of the CREB beta is not affected. Increased carbohydrate metabolism generally activates CREB alpha and beta, where after the activation, the CREB beta binds to its own promoter as a transcription factor, and it results in the production of even more beta, CREB beta isoform. CREB beta isoform serves as a marker of the CREB activity because of acute increase in the CREB beta after the CREB alpha activation. So this seems to be a very good marker. Increased hepatic CREB expression, which means both the expression of alpha and beta isoforms, have been reported in diabetes, type 1 and type 2 diabetes, obesity, and metabolic syndrome in humans, which indicates its potential as a diagnostic marker of metabolic status or metabolic syndrome. The current study was designed to assess the CREB expression of both of its isoforms in a hyperglycemic environment. This hyperglycemic environment in these rats were created by pre-treating the rats within. Alloxan is a chemical which induces significant beta cell damage, and this beta cell damage do not permit or do not let the pancreas to produce enough of the insulin. So the plasma or the blood level of these animals, a condition known as hyperglycemia. So the material methods, the next section is the material methods. 21 adult male rats were selected for this specific study. These animals were treated with a single intraperitoneal dose of Alexon at the dose rate of 120 milligram per kg body weight. The glucose levels in these animals were monitored through tail vein by glucometer. Because this study was the temporal assessment, which means that we are more interested in the timestamp of the expression pattern of crab. So these animals were sacrificed on multiple days. So these animals were sacrificed on the 0, 1, 3, 5, 8, 12, and 15th day. 0 day is the day on which the Alexon treatment was given, and the 1, 3, 5, 8, 12, 15 day are the post-Alexon treatment days. A total number of 3 animals were sacrificed on each of these days. After the sacrifice, the liver tissue was dissected and collected. Half of the tissue was placed in 4% formalin for histological assessment, whereas the rest of the tissue was placed in a chilled RNA letter solution for the RT-PCR expression. This expression reaches its maximum level after the 5th day. The expression pattern to the 8th day onward. On the other hand, the expression pattern of the beta isoforms gives us a little bit of a different picture. The initial phase, up till the 5th day, the pattern is kind of the same as the alpha, but after the 5th day, the significant decline which we observed in the alpha isoform is not there in the beta isoform. So this beta isoform, the transcriptional activity of this beta isoform does not seems to decline even in the presence of low glucose levels on the 12th or the 15th day, which means that this isoforms might be responsible for the glycolytic and the lipogenic even at the low glucose level. The other aspect of this hyperglycemia is that the high glucose level eventually induces reactive oxygen species, commonly known as ROS. And these reactive oxygen species, they eventually causes mitochondrial stress and endoplasmic reticulum stress. The mitochondrial stress and the ER stress eventually activates the antioxidant response elements in the genome. The major antioxidant response element genes are the NRF2 and NFKB. In order to observe the expression pattern in response to the high glucose level, we observe the expression pattern of the NRF2 and NFKB. Surprisingly, the expression pattern of NRF2 was quite different from the expression pattern of NFKB. So what we expected was that on the 3rd and 5th day, the expression levels will be high, which was the case as in the NFKB expression. But on the 8th day and 12th day, the expression pattern of these genes were still high. Glucose levels were low. So let's discuss these results. Hyperglycemia was achieved within 24 hours of the single Alexon dose and lasted for 8 consecutive days. From 8th day onward, downward trend in the blood glucose was observed, reaching almost normal glycemia. Expression profile of the crab isoforms affirms that high glucose concentration induce upregulation of expression. Hyperglycemia ultimately induced reactive oxygen species with further results in the mitochondrial and endoplasmic reticulum stress. So the mitochondrial and ER stress will eventually activate the antioxidant response elements. This is indicated by the sudden increased expression of NRF2 and NFKB. The timestamp or the temporal pattern of expression reveals that the crab expression represents a swift response towards hyperglycemia as compared to antioxidant response genes like NRF2 and NFKB. The subsequent decline in the crab isoform expression on the 12th and the 15th day might be attributed to the decline in the glucose level observed in these days, but that decrease was only observed for the alpha isoform. Activation of NRF2 and NFKB on the 5th day onwards can only be explained by the fact that substantial cytoplasmic concentration of ROS, reactive oxygen species, because of hyperglycemia was not achieved on the 1st or 3rd day of Alaxon treatment, which means that the high level of glucose needs some time to eventually produce the reactive oxygen species, and these reactive oxygen species need a little bit of time to eventually induce the mitochondrial or endoplasmic reticulum stress. So this time period during the 3rd day and the 5th day, where we don't see an increase in the NRF2 and NFKB expression, might be due to the fact that during this time the mitochondrial or the endoplasmic reticulum stress was not induced. So, but again, further data is always necessary to completely understand this complex mechanism of antioxidant response elements and the carbohydrate response element binding protein. Thank you very much. If you have any question, I hope I will be able to answer your question during the actual live online session. Thank you very much.
Thank you, Dr. Shahzad Irfan, for such an appreciable talk. Our 6th presenter of this session is Dr. Azhar Rafiq from Government College University, Faisalabad. She will present her research work on diversity and distribution of avian fauna in relation to urban landscape in Faisalabad city. Please, Dr. Azhar Rafiq from Government College University, Faisalabad.
Bismillahirrahmanirrahim. I am very thankful to this International Conference on Applied Zoology 2022, which is organized by the Applied Zoological Society of Pakistan, Khwaja Farid University of Engineering and Information Technology, Rahim Yarfan, Punjab, Pakistan. I am Dr. Azhar Rafiq, Assistant Professor, Department of Zoology, Government College University, Faisalabad, Pakistan. The topic of my presentation is diversity and distribution of avian fauna in relation to urban landscape in Faisalabad city. First of all, the introduction. Birds are great bioindicators of ecosystem because they are diverse and visible component of the ecosystem. Urbanization along with climate change is one of the most serious dangers to biodiversity, including the survival of many bird species. Fragmentation and habitat loss are perhaps the most pressing threats, causing people to make judgment calls about whether to migrate more suitable areas or they stay there and cope with the current situation. The surviving and existing green areas are modified through plantation of controlled lawns, non-native plant species, and loss of the mid-story canopy, resulting in the novel urban setting. The habitats of many animals, especially birds, have been affected as a result of the continued growth of civilization and urbanization. The degradation of birds' habitat has resulted in the extinction of several bird species. In recent decades, numerous studies have focused on the negative consequences of anthropogenic noise on avian diversity. Roads have a variety of harmful consequences on bird population. These effects have raised mortality rates. The current study was planned to determine the diversity of bird species in settlement areas and compare the bird diversity in urban areas with the other important ecological landscapes of the city. Interiors and method. The study area was classified into two major types, such as open fields and build-up areas. The major open fields include included the cemeteries, wasteland along canals and street banks, while the built-up areas included business centers, settlements, residential neighborhood. Data collection from September 2021 to May 2022. From September 2021 to May 2022, data was collected within plots using the point count method. The point count sampling design was consisted of a series of points at which birds were counted within a defined radius. 105 point count stations at 300-meter interval was established within several habitats. 5 plots randomly selected for each micro landscape within 5 into 5 kilometer grid. Each plot was visited for 20 minutes with the observation period during the day at dawn and dusk for 2 hours. Linear count method was used to determine the event diversity along the canals. Naked eyes, binocular and camera were used to count and photograph the birds. For identification of bird species, various practical guides were consulted. We also assessment of the vegetation. The vegetation assessment variable was sampled in these areas by using the 21 quadrant plots. In each plot, samples were recorded: soil cover percent, species richness, vegetation type, vegetation height, vegetation diameter, and microclimate. And the statistical analysis, for statistical analysis, version 3 was used to find diversity Shahzad index, where H is the Shahzad index, having a formula of this. Simpson index is used to estimate the dominance of the species and has no erroneous species richness. And the Simpson index formula is this one. Then the results. First of all, abundance. In current study, we recorded 26 species of birds of 22 genera from 17 families representing 13 orders. Peciformes was the dominant order representing 14 species and 10 genera and 17 families, followed by the Comparative with 7 species, 10 genera and 5 families. The total number of birds seen during the study were 1,649. 21 species plants were noted, for example, eucalyptus, ficus, and neem, etc. In terms of dominant, the order Pelicaniformes were ranked 3rd, while the dominant order was the Peciformes. The order which is 3rd one, Pelicaniformes, represented 3 species and 1 family. The Columbiformes order had 1 family and 2 species. Other orders, Apodiformes, Cetaziformes, Cuculiformes, Trigiformes, and Seriformes, etc. have 1 family and 1 species. House crow, a species of family Corvidae, remained dominant at all the study sites and had 517 number of birds. While the house sparrows, Passer domesticus, a species of family Passeridae, was on 2nd number with 222 numbers of birds. Common myna, a species of Pernidae, was 3rd in rank with the 176 number of species. The pie graph which represent the highest number of orders, which show the order of the different the birds. The order Passeriformes was the dominant, then Paradiformes, then all other orders. Now this table shows the association of birds with landscape and seasonal variation. A highly significant difference was observed in open field areas and in built-up areas. More diversity was found in open area, that is in canal ways, cemeteries, wasteland, and stream banks, while the enclosed area, business centers, settlement area, built-up areas have low diversity. Similarly, total number of bird species which were identified in both open area and built-up area were also very different. These difference also shows difference in the winter, spring, and summer. Winter and spring season shows the higher diversity as compared to summer. This table shows the structural properties of bird species for bird at different landscape in urban areas. These are the different landscape, canal ways, cemeteries, wastelands, stream banks, business and settlement area, and business centers. Business centers shows the lowest number of population of the bird species, as while the highest diversity was found in cemeteries and canal way. Similarly, dominant species were found, highest number were found in the cemeteries, then canal way, and lowest number was found in the business centers. This table shows the status of the birds in different seasons with their diversity index and evenness. The diversity index was higher in winter and spring as compared to summer. Also, number of species was higher in winter than summer, spring, and then summer. Then the conclusion, according to habitat fitness, bird species and numbers are higher in open area, which provide free water and abundant food and nesting opportunities. Plantation had an impact on birds, as well as there were more plant in open areas, resulting in a greater diversity of species. During winter and spring season, more birds were recorded because of its diverse species and food types. The order Passeriformes was the most prominent. Even though festivals have rich avian diversity due to variety of habitat and seasonal changes, but according to local reports, no previous study in this area exists. The birds' abundance and diversity fall down, especially in last few years. Population explosion may be the one major cause for decline of avian diversity. Thank you for your attention.
Thank you, Dr. Azhar Rafiq, for such an astounding and amazing presentation. Our seventh presenter of this session is Maria Amin from Quaid-e-Azam University, Islamabad. She will share her research work about livestock poisoning neem seed oil, a prospective feedstock for eco-friendly biodiesel production. Please, Maria Amin.
Dear researchers, I am Maria Amin, PhD scholar in Department of Plant Sciences, Quaid-e-Azam University, Islamabad. The topic of my presentation is livestock poisoning neem seed oil, prospective feedstock for eco-friendly biodiesel production. First of all, introduction. Due to rapid depletion of finite fuel resources, the exploitation of renewable energy such as biodiesel has become indispensable. More than 95% of biodiesel is produced from edible oil. However, due to its competition with the food supply, the non-edible feedstock is preferred. Let me discuss what is biodiesel. Biodiesel is basically defined as monoalkyl esters of long-chain fatty acids, and it is considered as petroleum diesel replacement fuel. Biodiesel is biodegradable, sustainable, renewable, non-toxic, with little or no sulfur oil content. Biodiesel feedstock. Biodiesel is basically produced from animal fat, edible or vegetable oil, non-edible oil, and different resources, for example, algae, fungi, bacteria, and waste cooking oil. Biodiesel feedstock across the globe. Biodiesel is mainly produced from rapeseed oil 84%, sunflower oil 13%, palm oil or soybean oil 1%, and other resources 2%. Now move towards neem seed oil. Neem seed or neem is botanically known as Azadirachta indica that belongs to family Meliaceae. It yields 40 to 50 kilogram fruits per plant each year. Neem seed oil has 69.4% unsaturated fatty acids, whereas 29.3% saturated fatty acids. Then material and methods. The titanium oxide catalyst was produced by co-precipitation method. The sample was oven dried and then calcined in the muffle furnace at 500 degrees Celsius for 3 hours simultaneously. The oil content was determined by using Soxhlet apparatus, and then FFA content was also determined in the neem seed oil. The biodiesel was produced by two-step methods. First was esterification and the other one was transesterification. And the two-step biodiesel synthesis process was followed in order to reduce the high FFA content of the neem seed oil. And as the result of this transesterification, fatty acid methyl esters were produced. The biodiesel that was impure, it was water washed and then it was purified. Then results and discussion. The highest biodiesel yield was achieved at 1:6 oil to methanol molar ratio, 0.1% of the catalyst loading, 60 degrees Celsius reaction temperature, and 3 hours of reaction time. FTIR analysis was performed and it confirmed that the methyl esters in the biodiesel have been achieved. GCMS of neem seed oil depicted that the occurrence of 14 different fatty acid methyl esters. Fuel properties were found in full accordance with the ASTM, that is International Standard. The flash point was found as 91 degrees Celsius. The pour point was minus 13 degrees Celsius and the cloud point was minus 10 degrees Celsius. Here we can see that the sulfur content is too much low or negligible, that is 0.00431 weight percent. And this is the reason that the biodiesel is renewable and non-toxic, and it is eco-friendly as well because it has too less or negligible amount of the sulfur in it. The total acid number was also found and it was also very. Then we move toward conclusion. The highest proportion of the biodiesel, that is 95%, was achieved using 1:6 oil to methanol molar ratio, 0.1% titanium oxide. Reaction temperature was 60 degrees Celsius and 3 hours of the reaction time. FTIR and GCMS confirmed the existence of methoxyester carbonyl group. Then fuel properties were found in full agreement with the ASTM standards. Neem seeds, being poisonous to the livestock, can be best feedstock for the biodiesel production. Then future recommendations. It is recommended that effect of phytonanocatalyst can be checked. The studies can be conducted in order to find out the efficiency of biodiesel with different blends by using phytonanocatalyst, and steps must be taken in order to improve the quality of the biodiesel. Neem seed oil cake. can be processed or detoxified to be used as animal feed. Thank you very much for your kind attention.
Thank you, Amin, for presenting your kind and informative research work at ICAZ-2022. Our eighth presenter and last presenter of this session is Malik Faiz Hussain from Bahauddin Zakariya University, Multan. He will share his research work on nitric oxide nanoparticles has the potential to drastically affect the behavior of albino mice in a sex-specific manner. Please, Malik Faiz Hussain from Bahauddin Zakariya University, Multan.
Bismillahirrahmanirrahim. I am highly thankful to ICAZ 2022 Conference of Applied Zoology. I am Malik Faiz Hussain, PhD scholar, Institute of Zoology, Bahauddin Zakariya University, Multan. My research topic is nickel oxide nanoparticle has the potential to drastically affect the behavior of albino mice in a sex-specific manner. First of all, the introduction of this topic. Nickel, a metal which is non-essential for human and mammal, but we are continuously exposing to this metal by various materials, pollutants from food and water. This nickel is continuously biomagnifying in our body, in the body of living organisms. The nickel metal interact with the cellular membranes as well as it can bind to the proteins and DNA and affecting the central dogma and gene expression profile. It can also be deposited in various organs such as lungs, skin, kidneys, and other vital organs of the body and approaching to the toxic and even carcinogenic levels. The nickel as a bulk is toxic, but the nickel oxide nanoparticles, which are nanotimes smaller to the bulky nickel as compared to the bulk nickel, so it seems that they can approach the various parts of cells and various vital organs easily and may cross the blood-brain barrier. In various research papers, nickel oxide nanoparticles in vitro studies are found toxic to the mammalian cells as well as they were proved toxic in lungs and other body parts also. So the current investigation aimed to report the effect of various doses of nickel oxide nanoparticles on neuromuscular functions, exploratory behavior, and object recognition capability of albino mice in a sex-specific manner. The methodology of this albino mice 5 weeks old C55BL6 strain were used and maintained under standard conditions in animal house of Institute of Zoology as the conditions already published in Hussain et al. 2020 different behavioral tests, Ratarod, open field test, light and dark box, and novel object recognition capability test were conducted after 14 days of intraperitoneal exposure of NIONP. The data collected as mean plus minus standard error of mean and were analyzed in Minitab version 17. The ANOVA and Tukey's post hoc test was used to compare the performance of various behavioral tests between experimental subjects and controls. The result of this exposure was found non-significantly on the body weight of albino mice in both genders, while Ratarod one-way ANOVA results revealed that the male mice injected with both doses of NIONP, low and high doses, the rotating drum staying ability of albino mice were significantly less as compared to saline injected male mice, while female mice were unaffected in Ratarod ability. Light and dark box test. One-way ANOVA results demonstrated that various parameters, only one of them is found statistically significant, which was stretch attend reflex. Open field test ANOVA results find that the mobile immobile episodes were significantly different from the controls in high dose in females. Novel object recognition trial 1 and trial 2 demonstrated that the approaching to the novel object were significantly different from experimental animals and was low as compared to the controls in female mice. In conclusion, we are reporting the drastic behavioral effects in mice following intraperitoneal injection of NIONP for 14 days. They affect the muscular activity, object recognition capacity, exploratory behavior of albino mice. The effects were more pronounced at higher doses while less pronounced in low doses. I am acknowledging this work to my mentor, Dr. Farhan Iqbal, who made me confident, trained me at every step. Thank you for all.
Thank you, Malik Fiaz Hussain, for such an informative talk of this session. Now it's time to end this session. I hope you all have enjoyed this session by amazing abstracts from different universities. Now it's time for the next session, which will be moderated by Ms. Komal Riaz, and it will include talks from the fields of natural products, COVID-19, and epidemiology. So stay tuned and connected with us. Thank you.